Good afternoon. I'm Dr. Jeffrey Hayes, Education Specialist for the American Society for Reproductive Medicine. Welcome to the latest presentation of the ASRM Grand Rounds webinar series. These webinars are designed to address topics in the ABOG Guide to Learning in Reproductive Endocrinology and Infertility. Today's presentation will be given by Dr. Mario Ascoli. Dr. Ascoli is currently Professor of Pharmacology and Professor of Obstetrics and Gynecology at Carver College of Medicine at the University of Iowa. The title of his talk today is Mechanism of Action of Protein and Peptide Hormones. I will now review the details of today's presentation. After the webinar is done, please do not forget to return to ASRM eLearn to take the post-test and complete the survey for your continuing education credits. You must complete the post-test question successfully and complete the survey to receive credit and be able to print off your certificate. If you wish to ask a question to the speaker about this presentation, when you return to ASRM eLearn, click on the page link labeled Questions, and an ASRM email address will be provided. After the time period for questions has expired, the questions page will be a frequently asked questions page pertaining to this presentation and topic. We're very excited for our speaker today, so I will now turn things over to Dr. Ascoli.
Okay, so what I am going to do today is discuss some of the different mechanisms of action of several protein and peptide hormone receptors as they relate to the function of the hypothalamic pituitary gonadal axis. I have no disclosures to make, and the objective of this lecture is basically to understand the molecular basis of the actions of three types of polypeptide hormone receptors on reproductive functions. This slide is just an introductory slide to remind everyone of the hypothalamic pituitary gonadal axis and some of the hormones that work on it. So, of course, kisspeptin is the hormone that is at the top, and it works on the hypothalamus to stimulate the synthesis and release of GnRH, that then works on the gonadotropes in the pituitary to stimulate the synthesis and release of LH and FSH, that then stimulate the testes and the ovaries to make androgens in the testes and progestins and estrogens in the ovaries. There are other actions, of course, such as spermatogenesis and ovulation. And the actions of androgens and progestins happen in, of course, many target tissues, including the hypothalamus and the pituitary, where androgens in the male and progestins and estrogens in the female have a feedback inhibitory effect that maintains the system in check. So these are the main hormones that everyone knows about. There are, however, a few others that are important. One of them is activin, which is a hormone that acts in an autocrine or paracrine fashion in the pituitary and in the ovaries. We also have another hormone of a similar nature, which is called inhibin, which is made in the ovaries and then feedbacks on the pituitary to control the synthesis and release of FSH. And then within the ovaries themselves, there are a number of hormones that are important to their function that we will discuss in this lecture, including epidermal growth factor, or EGF for short, ET, which is short for endothelin, CNP, which is short for the C-type natriuretic peptide, and PGs, which are short for prostaglandins. There are at least three different types of hormone receptors that are involved in signaling in the hypothalamic pituitary gonadal axis. One of them is what I will call the 7 transmembrane receptors, also known as G-protein coupled receptors, and does include the receptors for kisspeptin, GnRH, LH, and HCG, FSH, the 2 endothelins, the 2 prostaglandins, as well as GPR3 and GPR12, which are present in the oocytes and maintain the oocyte in meiotic arrest. There are also receptors that have an intrinsic enzymatic activity, and thus include receptors such as the Egf receptor, which have a tyrosine kinase activity, the NPR2 receptor, which is a guanylate cyclase, and then inhibin and activin receptors that have serine and threonine kinases. And of course, there are also receptors for steroid hormones, but we will not deal with those at all because those are not peptide hormones. So of the peptide hormone receptors, I'm going to talk about the 7 transmembrane receptors, and I am going to talk about 2 classes of the receptors that have intrinsic enzymatic activity, the Egf receptor and the NPR2 receptor. I will not cover inhibin and activin receptors. We'll start by talking about the 7 transmembrane receptors. To talk about that, we have to first talk about G proteins. These receptors were initially named G protein coupled receptors, and that's still a name that sticks around, but there are other ways by which they signal that we will discuss today. But we'll start with their ability to activate G proteins. So G proteins, as the name implies, are proteins that can bind guanine nucleotides. And when they bind GDP, they are in an inactive form, and when they bind GTP, they are in an active form. And the conversion of the inactive to the active form is actually facilitated by a family of proteins that are collectively known as guanine nucleotide exchange factors. The sole function of these guys is to actually promote the exchange of the GDP that was bound to the inactive protein for GTP that is present in the cell. And as a consequence of this exchange, then the G protein becomes active. Then it returns back to the inactive state because the GTP is hydrolyzed. These G proteins also have an intrinsic GTPase activity that is capable of hydrolyzing the GTP back to GDP. But often that hydrolysis is also aided by another group of proteins that are called GAPs, or GTPase activating proteins. Some of the G proteins that you recognize are, for example, RAS. This is a family of the small G protein family. There are others that are listed in here. And the G proteins that I'm going to talk about are the so-called heterotrimeric G proteins, because these are the ones that are actually activated by the 7 transmembrane receptors, which I will probably call 7 transmembrane receptors sometimes, and G protein coupled receptors other times. And there are 4 types of the heterotrimeric G proteins that are listed in here. We'll come back to that in a minute. So these are more complex in nature, and they're actually composed of 3 subunits designated alpha, beta, and gamma that are joined by non-covalent interactions. And the GDP is actually bound to the alpha subunit. When an agonist receptor complex, and here I'm talking about a 7 transmembrane receptor or a GPCR that is activated by an agonist, when that interacts with that heterotrimeric G protein, the function of this complex is to exchange the GDP that was bound to the alpha subunit for ambient GTP. As a consequence of that, then the subunits dissociate into an alpha subunit that now has GTP bound to it, and then a beta-gamma dimer. Beta-gamma always stay together, they never dissociate. The alpha subunit now that has GTP bound to it is actually an active unit that is capable of finding effectors that may be in an inactive conformation, and we're going to talk about that in a few minutes. And as a consequence of this binding, then the effector becomes activated. As I said, these G proteins have an intrinsic GTPase activity, and as a consequence of that, the GTP that is bound to the alpha subunit eventually becomes hydrolyzed back to GDP. And then the complex of the alpha GTP with the active effector dissociates, going back to the inactive effector. The alpha subunit that now has GDP bound to it is an inactive conformation, and then it finds beta gamma that were released up here, and then it goes back to the resting state. As we now know, beta gamma are also capable of binding to effectors in their own right, and these effectors may be the same or may be different effectors than those activated by the alpha subunit. And as a consequence of this interaction, then that effector becomes activated. Eventually, this dissociates just by the simple law of mass action, going back to the inactive effector and to the beta gamma that goes back to form the inactive complex that we started with. Now, the classical G protein-dependent pathway that you all know about is the pathway that leads to the stimulation of adenylate cyclase and the formation of cyclic AMP. And this pathway is actually activated by the alpha subunit of the so-called GS, heterotrimeric G protein. And as a consequence of this activation, there is an increase in the synthesis of cyclic AMP, and this is what is known as a second messenger. Cyclic AMP has a number of actions in the cells. The best known action is its ability to activate the cyclic AMP-dependent protein kinase, as shown in here. Cyclic AMP can also interact with cyclic AMP-gated ion channels, as shown in here. And it can also activate yet another family of G proteins by acting on this cyclic AMP-dependent guanine nucleotide exchange factor. The best characterized actions of cyclic AMP are actually through the cyclic AMP-dependent kinase. And then ultimately, of course, cyclic AMP is degraded by cyclic AMP phosphodiesterases going back to 5' AMP. There are, as I mentioned at the beginning, 4 different families of G proteins. GS, GIO, GQ11, and G1213. Don't worry about the meaning of these names because now they don't really mean anything. They just have some historical significance. And these G proteins can be activated by a variety of receptors, some of which are listed up here on the top. And if you look at the color arrows, you will see, for example, that a given receptor can activate more than one family of G proteins. If you concentrate on the Lh receptor, for example, this figure shows you that it will activate GS, and it also shows you that it activates GQ11. And we will come back to that later in the lecture. As a consequence of the activation of these G proteins, there is changes in the activity of metabolic enzymes, there is changes in transcription, for example, and then these changes lead to changes in the behavior of the cells, such as glucose metabolism, for example. Steroid production in the adrenal and in the gonads is activated this way. Pituitary function is also activated this way. And ultimately, then we have organismic changes, including embryo development, gonadal development, learning and memory, and reproduction. Some of the best known heterotrimeric G protein effectors are listed here. The main effector for G alpha S is adenylate cyclase. We talked about that a little bit, but there are also some calcium channels that are activated by this alpha subunit. The alpha subunit of GI also may activate or deactivate adenylate cyclase. It also has an effect on calcium, potassium channels. The alpha subunit of alpha Q of GQ11 activates phospholipase C and may activate RhoA, and then the alpha subunit of alpha 1213 is also an activator of RhoA. As I said, the beta gamma subunits also have some effectors of their own. Some of the best known effectors are some forms of adenylate cyclase, the Pi3 kinase, and also phospholipase. So where this field actually is at this point is in structural biology. We know quite a bit about the three-dimensional structure of these proteins, the G proteins and the receptors, as you will see in a minute. And actually now we're learning about how these proteins interact with each other and how drugs may affect their functions. So let's start with the G protein. So this is the three-dimensional structure of a generic G protein, and they all look very similar, not exactly the same, but very similar. And this is an artistic rendition of how they actually interact with the membrane and the receptor. The receptor is not here, but the alpha subunit is shown in blue here, and then is this bluish molecule shown in here. And then the beta subunit is shown in this magenta color, as shown in here, and then the gamma subunit is shown in this reddish color, as shown in here. And these G proteins actually are peripheral membrane proteins in that they associate with the membrane through the fact that they have lipid modifications. And there's a mistake in this figure that I noted here in red. The lipid modifications here are wrong. The myristoyl moiety is actually in the N-terminus of the alpha subunit, and the geranyl moiety is actually in the C-terminus of the gamma subunit, where this is. And you can see here this little molecule shown in here, that's the GTP that is bound to the alpha subunit. We also know quite a bit about the structure of these receptors. And this is a prototypical structure of a G protein coupled receptor or a 7-transmembrane receptor. This happens to be the beta-2 adrenergic receptor, which is one of the best characterized receptors in this family. This is the agonist bound in here. So this is the outside of the cell, and this is the inside of the cell. And as you can see, there's some extracellular domains in here and an intracellular domain in here. And these receptors are characterized by the presence of these 7 transmembrane helices that are shown here in different colors. And that's where the name comes, 7 transmembrane receptors. So the N-terminus is in the extracellular domain, and then the receptor will weave in and out of this membrane 7 times to produce the 7 helices. And then the C-terminus is actually in the intracellular space as shown in here. There's an 8th helix that is intracellular. And this figure here on the right shows you the 3-dimensional structure of a receptor bound to a G protein. And this is as we know them. So this actually shows you the receptor associated with the inactive G protein. This shows you the receptor in the process of activating the G protein, and this is a superimposed inactive and active form of the G protein. And the main point to be taken from this figure is that there are some really substantial changes in the 3-dimensional structure of the G protein that happens when it becomes activated. And you can see it here if you compare the yellow and the blue colors. The yellow is the active G protein, and then the blue is the inactive G protein. And one of the most important changes is that alpha helical domain of the alpha subunit flipping from this position to that position. And there's also some other changes on the N-terminus that are not particularly obvious in here that are important as well. So there are in the neighborhood of 800 7-transmembrane receptors in the human genome. And there are at least 17 different alpha subunits of the G protein, 7 different beta subunits, and 12 different gamma subunits. Even if you assume that each 7-transmembrane receptor activates a single family of G proteins, and that is not the case, as I will discuss in a minute. And even if you assume that if each G alpha and each beta gamma activate a single effector, which is also not the case, the potential number of signals generated from a G protein activation is very large. And that actually gets even compounded by the fact that a given G alpha or a given G beta do not activate a single effector, which was what the initial thought was. A given G alpha or a given G beta gamma pair can and in fact do activate more than one effector. And if you go back a couple of slides, I gave you some examples of what effectors may be activated. Also, the initial thought that a given receptor may activate a single family of G proteins is also antiquated and wrong. In fact, most of these receptors can activate at least 2 families of G proteins, but there is selectivity or preference for one of the 2 families. So these considerations, when you think about it, make the 7-transmembrane receptor G protein system incredibly versatile and complex. And in fact, all physiological functions, or virtually all physiological functions, including reproduction, are dependent on 7-transmembrane receptors. In addition, there are estimates that about 30 to 50% of all the drugs that are used in clinical pharmacology, including FSH for the induction of follicular growth and LH or HCG for the induction of ovulation, actually work on 7-transmembrane receptors. All right, so let's add a little bit of complexity to this. So this is what I told you the beta-2 adrenergic receptor looks like, with the 7 transmembrane helices. The agonist bound in the middle of this helices, a small extracellular domain, and then the intracellular domain shown in here. There's a subclass of G protein coupled receptors that, in addition to having this peculiar structure that they all do, also has this very large extracellular domain, as shown in here. And this large extracellular domain is characterized of the so-called lucid reach repeats, which form a peculiar structure that is depicted in here, but I will show it to you in real life in a few minutes. And I am mentioning this because the glycoprotein hormone receptors actually fall into this category. So the LH receptor, the FSH receptor, and also the closely related TSH receptor all have this structure depicted in here. And this is important because the ligands that they bind are rather large. For example, if you compare the ligand that binds the beta-2 adrenergic receptor, which is epinephrine, for example, is a rather small molecule compared to LH or FSH or HCG, which are rather large molecules. And that's the reason why these large extracellular domains have evolved. There are 2 other receptors that are important to reproduction that are members of this family. One is the LGR7 and the other one is LGR8. LGR7 is actually an LGR8 bind small molecules that are members of the relaxin family. LGR8, in fact, is a receptor for relaxin, and LGR7 is a receptor that is involved in testicular descent, and it binds this ligand called INSL, which is a peptide that is related to insulin that participates in testicular descent. So this is actually the 3-dimensional structure of the extracellular domain of the FSH receptor. This was solved a few years back. And if you look at this structure, this would be like the 7 transmembrane helices that I showed you before that are peculiar to all of these receptors, or common, I should say, to all of these receptors. And this, what I'm going to show you here, is this large extracellular domain that is present in the FSH receptor, the LH receptor, and the TSH receptor. And that is characterized by these lucid-rich repeats that are shown here in yellow. And the way that this looks in real life is like a half a banana, as you can see in here. And then there is this structure that it doesn't have a lot of order structure that connects this extracellular domain to the 7 transmembrane helices. And this actually shows the 3-dimensional structure of the extracellular domain of the FSH receptor, shown in purple here with the hormone bound to it, which is shown in gray, FSH. So it's actually like a hand clasp where the receptor then is one hand, as shown here, and then the hormone is the other hand, as shown in here. All right, so we talked about these receptors working through the activation of G proteins, and we discussed this particular mechanism by which they act, which is by activating a G protein. But during the process of activation, there's an additional kind of phenomena that becomes important. So after the process of activation, the G protein coupled receptors, or 7 transmembrane receptors, actually become phosphorylated in their intracellular regions, as shown in here. And this phosphorylation is catalyzed by a family of kinases collectively known as GERCs. And as a consequence of this phosphorylation, these receptors are now able to bind another important family of proteins that are known as the beta-arrestins. And one of the important features of the beta-arrestins is that it's a competitive binding with the G protein. So if beta-arrestin is bound to the receptor, the G protein cannot be bound to the receptor. In fact, this is why they're called beta-arrestins, because they were known to arrest the process of G protein activation. So this phenomena then serves to stop the action of the receptors by activating, because they stop activating the G proteins. And this has been known for a while. But more recently, it's become clear that in addition to just preventing the binding of G proteins, the beta arrestins are important signaling molecules of their own right. One of the consequences of the binding of arrestin to the receptors is that the receptors are targeted to clathrin-coated pits where they are internalized into the cell. And this is another reason why these receptors then stop working, simply because they're not in the right location anymore. But another very important aspect of this is that the beta-arrestin can bind a number of other signaling molecules, such as SRC, for example, AKT or ERK. These are important kinases that activate different processes in the cells. And then as a consequence of this, the formation of this complex, then there's an additional set of signals that is now dependent on beta-arrestin as opposed to G proteins that is important in the action of these 7 transmembrane receptors. And this is the reason why people want to change the name, because they're really not G protein coupled receptors anymore. It's true that they are, but it is also true that they now work through this beta-arrestin pathway. This is really an important concept that really hasn't really paid too much, it doesn't really have too much of a function in reproductive processes for the time being, and that may be simply because we don't know much. But it may turn out to be important. It is certainly important in the action of certain drugs that work through other receptors, such as the adrenergic receptors or the angiotensin receptor. So this is the concept. The idea initially was that when an agonist binds to a receptor, then it will act in a similar manner, either through a G protein or through the arrestin pathway. And this is what you will call balanced signaling. But it is becoming clear that that is too simplistic. I mean, there are actually agonists that will actually work preferentially through the beta-arrestin pathway, and this is called then a beta-arrestin biased agonist. And there are other agonists that work preferentially through a G protein pathway, and this is then called a G protein biased agonist. And carvedilol, which is used in the treatment of cardiac disease, is perhaps the best documented example of a drug that works on the, through the beta-arrestin pathway rather than G protein pathways. By the same thinking, there may be receptors that are biased either through by working through the beta-arrestin pathway or through the G protein pathway as well. And a lot of the thinking about the development of drugs and how they may act is actually based on this paradigm that I've shown you here. But as I said, right now we don't have a good example of how this may work in reproduction. But that, again, may be because of our ignorance. So then I want to close the G protein coupled receptors, or 7 transmembrane receptors, by coming back to their importance in reproduction. So some of the most important G protein coupled receptors involved in reproduction are here, and then there's clear that these are involved in reproduction functions. The receptor for Kispectin is a member of this family. The receptor for GnRH is a member of this family. The LHCG receptor is a member of this family, and the FSH receptor is a member of this family. And there are a number of diseases that are associated, that are due to mutations of this receptor, these receptors. And these are diseases, the ones that I listed in here, that are due to mutations that that prevent these receptors from functioning well. So this would be a mutation that induces a simple loss of function of the receptor, but more often what these mutations do is they prevent the receptors, the newly synthesized receptors, from traveling from the intracellular space to the plasma membrane. So they're simply retained inside, and because of that, there is a hormonal refractoriness to the ligand because the receptor is not at the plasma membrane. All of these mutations are autosomal recessive in nature. So an inactivated mutation of the KIS1 receptor, the Kispectin receptor, is the cause of a disease called central hypogonadotrophic hypogonadism. The same can be caused by mutation and inactivated mutation of the G and Rh receptor. If there are inactivated mutations of the Lh receptor, this will lead to familial hypogonadism and Leydig cell hypoplasia in the males, primary amenorrhea in females. And likewise, inactivated mutations of the FSH receptor will lead to subfertility in males and ovarian dysgenesis in females. The other side of the coin are actually mutations that make these receptors think that the hormone is bound to them. In other words, these receptors are activated by the mutation rather than by the hormone, so they become independent of the presence of the hormone. They're active in their own right. And there are a number in here that are actually important in reproductive processes as well. There is an activating mutation of the KIS1 receptor that is causative of central precocious puberty. This actually was in a girl, and we don't know how it's inherited because the girl was adopted, so we don't know her family history. And the reason why this receptor actually is constitutively active is because it doesn't desensitize. In other words, it's unable to stop activating G proteins or arrestins for that matter. There are activating mutations of the Lh receptor. The phenotype of these mutations is actually male limited. There are women who have activating mutations of the Lh receptor, but they have no phenotype. So in males, it leads to male-limited precocious puberty or precocious puberty and Leydig cell adenomas. The male-limited precocious puberty are autosomal dominant mutations. The only mutations that are known to induce Leydig cell adenomas are actually somatic in nature, but they induce precocious puberty as well. And then there's a number of interesting mutations of the FSH receptor that broaden the specificity of this receptor, so it can now respond to TSH and respond to LH as well. And this results in ovarian hyperstimulation. All right, so we have discussed then the 7 transmembrane receptors, some of the aspects of their function and their importance in reproductive processes. And what I want to do now is close by discussing the receptors with intrinsic enzymatic activity. And there are 2 types of these receptors that I will discuss. One is the receptors that have a tyrosine kinase, and the Egf receptor is a prominent example of that. And another one is the receptors that have a guanylate cyclase activity, and the NPR2 receptor is an important member of that family. Both of these are particularly important in oocyte maturation, as you will see at the end of the lecture. So the Egf receptor is a receptor that is a member of the so-called ERB family of receptors. There are 4 of them. Egf receptor, also known as ERB1, and then there's 2, 3, and 4. They all have the same overall structure. So this is the extracellular domain in here. These receptors span the membrane only once, as opposed to the 7 times that the other receptors span the membrane. And then they have an intracellular domain that has an intrinsic tyrosine kinase activity. This tyrosine kinase activity, as the name implies, is capable of phosphorylating tyrosine residues, and the ligand binds in the extracellular domain. The ligands are somewhat different for these 4 different types of receptors. In terms of the Egf receptor, the most important ligands, and we will discuss this in some detail later, are amphiregulin or ER, EGF, TGF-alpha, epiregulin, beta-cellulin, and other forms of the Egf ligand that are depicted in here. Some of these ligands may bind to some of the other type of ERB receptors, as some may not. But all the important ligands for these 4 types of receptors are shown in this row over here. So this is a depiction of how these receptors work. So this is an artistic rendition of the Egf receptor, the Egf binding site in here, the single transmembrane spanning domain, tyrosine kinase activity, and then the cytosolic tail that has a number of tyrosine residues in it. When Egf binds to this receptor, one of the important things that it does is allows these receptors to dimerize. So you form these dimers of Egf receptor, and then the cytoplasmic tails come in close opposition to each other. And because there's a tyrosine kinase activity in this tail and a tyrosine kinase activity in this tail, they can transphosphorylate these tyrosine residues. And the end result is that you get a lot of tyrosine residues in the C-terminal tail, intracellular C-terminal tail of these receptors that become phosphorylated. What this does then, and this is another depiction of this process, this is Egf, and this is the two monomers of the Egf receptor in here, and then the P's are the different phosphorylated tyrosines. So what the different phosphorylated tyrosines do is they become docking sites for molecules that have activities of their own. An important one is Schick, which is a molecule that binds to this particular tyrosine kinase depicted in here. And then it forms this really large complex that is shown in here. And ultimately, the result of the binding of Schick to this phosphorylated residue is the activation of the ERK cascade, which you will know as being an important kinase cascade that modulates a number of transcription factors in different cells. Another important pathway that is modulated this way is the AKT and the Pi3 kinase pathway. This happens by the binding of one of the subunits of Pi3 kinase, P85, to another tyrosine that is phosphorylated. And then another important pathway that is activated this way is the inositol phosphate cascade, which is activated when an enzyme called phospholipase C gamma binds to yet another phosphorylated tyrosine residue present in that receptor. So that's basically how they work. These tyrosine residues form docking sites where different molecules can dock to and then become activated by proximity or perhaps by conformational changes. Now, this happens to be important because one of the consequences of the actions of LH in granulosa cells, when it activates GS and it increases cyclic AMP, one of the important consequences of that is that there is an increase in the synthesis of three of the ligands that bind to the Egf receptor. That's amphiregulin, epiregulin, and beta-cellulin. This happens, of course, in the mural granulosa cells when LH activates the LH receptor. And this actually is important in the vectorial transfer of information from the mural to the cumulus cells, because cumulus cells don't have LH receptors, yet they have to, one way or another, respond to the LH search. So what is thought that happens is when we have a granulosa cell, and granulosa cell in this case means the mural granulosa cell, when you have the LH receptor binding to, LH binding to the LH receptor, then you get an increase in the synthesis of amphiregulin, epiregulin, and beta-cellulin. And because the mural as well as the cumulus cells have Egf receptors, now these receptors in the mural cells and in the cumulus cells become activated as a consequence of the activation of the LH receptor. So the cumulus cells don't have to respond directly to LH. They do respond only indirectly because of this pathway that I depicted in here. And that actually is essential for cumulus expansion, as shown in here, and it's also essential for ovulation. We will come to that in a little bit. So the last important family of receptors that I want to discuss is the receptors that have an intrinsic guanylate cyclase activity. And there are several of them that are shown in here. The overall structure of these receptors is in many ways similar to that of the Egf receptor. They have an extracellular domain, they have a single transmembrane domain, and they have an intracellular domain. In this case, instead of having a tyrosine kinase activity like the Egf receptor did, these receptors have a guanylate cyclase activity. And as a consequence of the activation of these receptors, they convert GTP to cyclic GMP. So like cyclic AMP, which many of you know about, cyclic GMP is also a second messenger that can activate many pathways that I will describe in a few minutes. The ones that are important for reproduction are these two in here. The so-called PGC8, which is also known as NPR1, or PGCB, which is also known as NPR2. PGC stands for particulate guanylate cyclase, and NPR stands for the receptor for atrial natriuretic peptide. So the ligands for these receptors are A and P, which is the A-type natriuretic peptide, the B-type, and the C-type. And in terms of ovulation, the important players here are NPR2 and CNP, which is the ligand that activates NPR2. So this is the one that we're talking about, this blue one in here. But its overall structure is very similar to the others. So this now then shows you the type 2 NPR receptor. And this is in a generic cell, and there are actually phosphorylation sites in the intracellular domain of this receptor, and those phosphorylation sites, when phosphorylated, can affect the function of the receptor, as we will discuss in a few minutes. And in affecting the function, then it can affect its ability to make more or less cyclic GMP, as the case may be. But the main function of these receptors then is to make cyclic GMP. And as I said, cyclic GMP is a second messenger. And one of the important functions of cyclic GMP is actually to activate a family of kinases called cyclic GMP-dependent kinases, just like there are cyclic AMP-dependent kinases. Another important function is to activate the cyclic GMP-dependent channels that are shown in here. This is not particularly relevant to reproduction. This may be. Perhaps one of the most important functions of cyclic GMP in reproduction is its ability to inhibit PDE3, which is a phosphodiesterase that is capable of degrading cyclic AMP. And we will come back to that in a few minutes. So cyclic GMP then is also degraded in its own right by different phosphodiesterases shown in here, particularly PDE5, 6, and 9. And in addition to inhibiting PDE3, cyclic GMP can also activate PDE2 and affect the levels of cyclic nucleotides. So to put it in a nutshell, one of the important actions of cyclic GMP in reproduction is to actually modulate the catabolism or the degradation of cyclic GMP and actually also cyclic AMP as well. So the NPR2 receptor is very important in ovulation, and this has actually been described very recently in the past couple of years or so. And one of the important actions of LH turns out that it dephosphorylates some of these phosphorylation sites that are present in the NPR2 receptor. The NPR2 receptor is present in the mural granulosa cells. And then when LH activates the LH receptor, one of the important consequences of that is an activation of a phosphatase activity, leading to a dephosphorylation of these sites and then a decrease in the ability of the NPR2 receptor to make cyclic GMP. So you go from making a lot to making few, to making less cyclic GMP. In addition, as time progresses, LH also decreases the synthesis of CNP, which is the ligand for this receptor, leading to a further reduction in the activity of this receptor to stimulate the synthesis of cyclic GMP. And as you will see in a few minutes, this is all important in meiotic arrest and in ovulation. So let me try to put all of this together in the context of the three receptors that we discussed. So the three types of receptors that we discussed. So the Lh receptor is a prototypical G protein coupled receptor. This is present only in neural granulosa cells. The Egf receptor is a tyrosine kinase receptor and is present in neural and cumulus cells. NPR2 is a guanylate cyclase and is present mostly in the neural cells. And then we have GPR3, which is also a G protein coupled receptor that is present in the oocyte. And as you know, there's an interconnection between these three cell types using these tight junctions. Some of the tight junctions have connexin 43, others have connexin 37, as shown in here. So in the resting state, then you have very little cyclic AMP being made in the neural granulosa cell because the Lh receptor is not active. You have a lot of cyclic AMP being made in the oocyte because GPR3 is active. We don't know what the ligand for GPR3 is. Maybe there is no ligand. Maybe it's just active on its own. But there's a lot of cyclic AMP in the oocyte because of that. And also because the main phosphodiesterase that is present in the oocyte is PD3A, and this phosphodiesterase is actually inhibited by cyclic GMP. So the oocyte has high levels of cyclic AMP and high levels of cyclic GMP. And one of the important functions of the cyclic GMP is to inhibit the degradation of the cyclic AMP. Now, where does that cyclic GMP come from? There is no NPR2 in the oocyte. Well, so actually it comes from both the cumulus cells and from the neural cells. The neural cells actually make a fair amount of CNP, which is the ligand for NPR2, which is also present in the neural cells. So when there is a lot of CNP activating NPR2, then there will be a lot of cyclic GMP being made in here that can freely diffuse to the cumulus cells through these gap junctions, and that can freely diffuse from the cumulus cells into the oocyte through these gap junctions in here as well. So whereas the cyclic AMP present in the oocyte comes from the oocyte, the cyclic GMP doesn't come from the oocyte. It comes mostly from the neural granulosa cells. So this is the arrested state, right? And then as you know, an oocyte is arrested because of the high levels of cyclic AMP. So that's what induces meiotic arrest. So let's consider now what happens during the LH surge. So we'll start with the same condition, right? So high cyclic AMP, high cyclic GMP, and then very little cyclic AMP in here, and a lot of cyclic GMP in these compartments for the reasons I just discussed. So when the LH surge happens, there's a stimulation of the Lh receptor, and now you get a lot of cyclic AMP in the neural granulosa cells. One of the consequences of that is an increase in the synthesis of these three EGF-like growth factors that we talked about, amphiregulin, epiregulin, and beta-cellulin. And then this can activate the EGF receptors that are present in the neural cells, and also the EGF receptors that are present in the cumulus cells. The activation of the EGF receptor in the cumulus cells is essential for cumulus expansion. If you don't have this activation, cumulus expansion just doesn't happen. So this is then an important effect of the increase in the ligands for EGF growth factor that is caused by LH when it acts on the neural granulosa cells. And so another important effect of the activation of the EGF receptor on both the neural granulosa cells and the cumulus granulosa cells is the inhibition of the function of connexin 43, which is an important component of the gap junctions between the mural and the cumulus cells. And yet another important aspect of the function of the Egf receptor is that partial inhibition of the activity of NPR2 in the mural granulosa cells. So what do you think will happen with this, right? So if you inhibit NPR2, there is going to be less cyclic GMP that is going to be formed. But another consequence of this is an increase in the activity of PDE5, which degrades cyclic GMP. So when you consider then that there's an inactivation of the NPR2 receptor, there's an increase in the PDE5 activity, then that creates an environment where there is going to be less cyclic GMP being synthesized and more cyclic GMP being degraded. This is actually confounded by the fact that there's a decrease in the synthesis of CNP in the mural granulosa cells. So the NPR2 receptor becomes even less active. So the inactivation of the NPR2 receptor, which happens by a number of mechanisms that I just discussed, as well as the increase in PDE5 activity in the mural granulosa cells, lead to a decrease in the cyclic GMP that is being produced. And there's also degradation of cyclic GMP in the cumulus cells. And now, because of these gap junctions are closed, there is really no way for this cyclic GMP to flow between these two compartments. So what happens now is that the cyclic GMP that is high in the arrested oocytes will now flow into the cumulus cells, and that will decrease. And then the cyclic GMP will be degraded again in these two compartments, leading to an overall decrease of cyclic GMP in the entire system, but most importantly, a decrease in cyclic GMP in the oocyte. Because the cyclic GMP is essential to inhibit the phosphodiesterase that is present in the oocyte, that inhibition will go away. Because that inhibition goes away, more cyclic AMP is going to be degraded. That leads to a decrease in cyclic AMP and ultimately to the resumption of meiosis in the oocyte. So what I tried to explain in here is how three different types of receptors, the Lh receptor acting on mural granulosa cells solely, then the Egf receptor in mural and cumulus cells, and then the NPR receptor in mural cells, how the actions of all of these receptors ultimately affect the oocyte, which doesn't have Lh receptors, and lead to the resumption of meiosis and eventually the release of a fertilizable oocyte. All right, so let's talk a little bit more about this process of ovulation. And so we talked about ovulation in necessitating cumulus expansion and necessitating the resumption of meiosis. But another aspect that is needed is for those follicles to rupture so that the fertilizable oocyte can be released. And all of this, of course, is under the influence of LH acting only on the mural granulosa cells. And I discussed already some of the processes that lead to cumulus expansion and meiosis. And I'm not going to go through that again. The only thing that I'm going to tell you is that there's another important component of this pathway, which is the ERK1/2 cascade, that we know is part of this process, but I didn't really have the time to discuss it. And I will come back simply now to tell you that this is all mediated by cyclic AMP, which is made by the activation of GS in the mural granulosa cells through the Lh receptor. What we also know now is that in the mural granulosa cells, the Lh receptor can activate another family of G proteins, which is called GQ11. And the co-activation of GQ11 and GS, neither one of them is enough to elicit the full process. The activation of these two is necessary for the induction of the progesterone receptor in the mural granulosa cells. That will ultimately then work to increase the synthesis of ADAMTS and catepsin L and perhaps other enzymes as well that are essential for the rupture of the follicles. So the three processes then, cumulus expansion, the meiosis resumption, and then follicular rupture that are necessary for ovulation are all under the control of the LH receptor and two different families of G proteins, GS and GQ11. So these are the conclusions, the overall conclusions from the lecture then. So activation of the Lh receptor, a 7-transmembrane receptor that is present in mural granulosa cells, is essential for the ovulatory response. There's no question about that. Everybody knows that. The actions of the Lh receptor are mediated by the activation of two families of G proteins, GS and GQ11. The activation of GS is necessary for the inhibition of NPR2 activity, the increased degradation of cyclic GMP, and the increase in the synthesis of EGF-like growth factors in mural granulosa cells. The increased levels of EGF-like growth factors then activate the Egf receptor both in cumulus cells and in mural cells. But importantly, in the cumulus cells, the activation of this receptor is necessary for cumulus expansion. Then the decreased levels of cyclic GMP in the mural granulosa cells ultimately result in a decreased levels of cyclic MP in the cumulus cells and the oocyte. So these decreased levels of intraoocyte cyclic MP is what allows for the resumption of meiosis. So the activation of GS and GQ11 are both required for the increased expression of the progesterone receptor in mural granulosa cells. And the increased expression and activation of the progesterone receptor, remember that progesterone is high at the time of ovulation, are essential for follicular rupture. Then ultimately, ovulation of a fertilizable oocyte requires the activation of one 7-transmembrane receptor, which is the Lh receptor, two families of G proteins, GS and GQ11, a tyrosine kinase receptor, the Egf receptor, a guanylate cyclase, which is the NPR2 receptor, and a steroid hormone receptor, which is the progesterone receptor. And the Lh receptor and the progesterone receptor are expressed exclusively in mural granulosa cells. NPR2 is expressed mostly in the mural granulosa cells, a little bit in the cumulus cells. And the Egf receptor is expressed in both mural and cumulus granulosa cells. And that is all I have to say. Thank you.